Thanks for the introduction. This is a megakaryocyte. As you can see in this family tree, they originate from pluripotent myeloid stem cells, which are found in bone marrow and produce other blood-related cells. I say other because these megakaryocytes are the source of the platelets we find in our blood. Without these non-nucleated thrombocytes, we wouldn't be able to prevent and stop bleeding, and would definitely die. These megakaryocytes make platelets by trying to divide via mitosis, but stopping midway. This process happens a couple more times before the megakaryocyte is fully matured, and by the end of it, it can have up to 200 chromosomes in its lobulated nucleus. That's why it looks so strange. It also gets bigger after every failed division, which can end up with it having a diameter of 0.1 millimeters, making it 10 to 15 times bigger than a red blood cell and visible to the naked eye. These massive guys, at the end of their lifespan, will try to make their way to bone marrow sinusoids or capillaries and try to force their way inside. This inevitably doesn't work since they're huge, so only small bits protrude into the blood vessel. These protrusions are then hit with high pressure from the blood and get torn off until the megakaryocytes are gone and thousands of platelets have been formed. Kind of like a backwards cheese grater. The real question, however, is what is with this halting of mitosis? Well, a couple of scientists in the American Society of Hematology in 1998 had the same exact question. Vitrat et al. Wanted to see what this endomitosis was caused by. In order to do this, they needed a lot of megakaryocytes. This is a problem, since there aren't many in the body at one time. To get over this hurdle, they got a large sample of human megakaryocytes by getting some blood and then adding a type of marker called CD34, which isolates bone marrow precursors, or hematopoietic stem cells, which are stem cells capable of forming into megakaryocytes. They then added these stem cells into a vial and added MPLL, or thrombopoietin, which stimulated the megakaryoblasts to be formed. Now, with their multitude of massive cells, they look deeply into their various cell cycle sections. Up until the cell's start of the chromosome separation phase, or metaphase, everything seemed fine, besides some strange-looking circular spindles. These deformed spindles are still able to pull the chromatids apart, but only slightly. The cell cycle continues as normal, but since the chromatids are too close together to form two different nuclei, one nuclear envelope is formed around them, creating a lobulated nucleus. As a consequence of this, the cytokinesis phase, which is the final separation of the two cells, acquires a major defect, causing it to abort its processes and induce mitotic exit. This is the final result that the study concluded. Although this was only a small portion of what the paper talked about. Much of the paper was catered to adding evidence to debunk 3 other theories, all of which I didn't feel need to include, due to them adding no necessary context for the actual findings. Although one of these theories, that an absence of one of the enzymes required for cell division, called cyclin B1, caused endomitosis, was what made the researchers test for the enzyme specifically, in which they found throughout the cell cycles of the megakaryocytes, so it proved the hypothesis faulty. But their focus on cyclin B1 had them find that it seems to play a role in the creation of abnormal spindles. They found that an abnormality in the degradation of cyclin B1 could partially explain many of the aspects of endomitosis, including the spindles. Now, what are the implications of a study like this? Well, for one, the more we know about molecular mechanisms that pertain to cells that are important for bodily function, the better. But it also gets us closer to finding ways to help people with thrombocytopenia, or low platelet counts. If we use what we know about how endomitosis works, maybe we can better control it and help people with this potentially fatal condition.